Okay, I think we can start. We have 25 people and more people will contact, I'm sure. So it's my pleasure to have today for the monthly webinar of EUROTOX Professor François Huaux. So François Huaux is the head of Louvain Center for Toxicology and Applied Pharmacology, which is at the Catholic University of Louvain in Belgium. He is a professor of food and chemical toxicology. He's also editor-in-chief of the journal Frontiers in Toxicology, the immunotoxicology section, and vice president of the Belgium Society of Toxicology and Ecotoxicology, the BELTOX, and an active member of ETCAS, which is the immunotoxicology and chemical allergy specialty section, which is a specialty section of EUROTOX. And his current research program is dedicated to study the impact of macro and nanometric inhaled particles on the immune system and human health, with a particular focus on particle-induced chronic disease. So I'm very pleased, François, that you accepted to give this webinar because you are widely known for your expertise on the pulmonary toxicity and particle toxicity in the lung. So thank you very much. You will have 45 minutes. Then we have 15 minutes of questions. So please put your question in the chat, and then I will read it to François. The presentation is recorded, so you will find it on the YouTube channel on the EUROTOX website. And please shut down your video and microphone during the presentation. Thank you. François, the floor is yours.
Okay, thank you very much, Mark, for this presentation and the invitation. Today, I would like to start by presenting the goal of my research group. Okay, my lab tried to better understand diseases caused by inhaled toxic particles. This is my topic since many years. This slide has been part of my presentation for at least two decades. And it outlines a fundamental observation. Inhaled particles can lead to a wide range of diseases, from fibrosis and inflammation to cancer and autoimmune diseases. Particles are also known to amplify very diverse diseases such as allergies and infection. However, the exact mechanisms driving the diverse effect remain unclear. Our research focuses on the idea that exposure to particles such as asbestos, silica, and nanoparticles affects the immune system, and this immune system dysfunction is believed to play a central role in disease progression. You have here a beautiful picture of macrophage exposed to nanoparticles in red. When reviewing the toxicology literature, we find similar patterns. Both solubles and insoluble substances have the ability to trigger various diseases, and these effects are mediated through significant immune system disruption. And this concept aligns with the field of immunotoxicology, which suggests that toxic substances have both primary and secondary targets. Their impact on these targets can lead to inappropriate immune activation, and dysregulated immune responses are associated to adverse outcome. One key mechanism behind this simple view is immunostimulation or inflammation. Indeed, inflammation is a magic word used to explain everything in immunotoxicology. And you have here, I need the pointer now. You have here what I call inflammogenic toxicity. That means the different steps leading to adverse outcome. And now, in immunotoxicology, we know that toxic substances can directly activate immune sensor, and inflammasome activation. And this activation sensor and inflammasome can lead to what we call now sterile inflammation. And the sterile inflammation is dependent of immunogenic cell death induced by inflammasome activation, but also the release of cytokines, and the accumulation of numerous neutrophils. Mainly the neutrophils can induce mutation and induce secondary genotoxicity, and growth factors and cytokines released after immunogenic cell death can activate cells. Toxicity, genotoxicity, and cellular activation can lead adverse outcomes like chronic inflammation, cancer, fibrosis, and autoimmune diseases. I would like to precise that we know very well, and we describe very well AOP. I just present here an AOP for my group concerning silica particles, but it's the same for different toxicants and pollutants. The idea is the first event is often, inactivation of the cells. Here, silica can activate membrane and induce membrane permeabilization inside the cells after phagocytosis. This activation and mitochondrial stress will lead to the inflammasome activation. And probably everybody here knows that the inflammasome activation can lead to the activation of cytokines like I1, IL-30, 18. But the activation is necessary, of course, but the release also. And we know that there are some pores here on the membrane of macrophage activated by silica that allow the release of activated IL-1 alpha, IL-1 beta, and other cytokines. Importantly, all those pro-inflammatory cytokines released, activated, can induce the acute first, and then the chronic recruitment of neutrophils. And we think that inflammation, neutrophils, but other cells as well, can lead to fibrosis, cancer, and autoimmunity. So the AOP of inflammation in toxicology is very well known, well described, well defined. But today I would like to talk about a contrasting word, is immunosuppression. I will try to convince you that immunosuppression is very important also in the AOP, this view of the immunotoxicology. I would like first to well define inflammation versus immunosuppression. For me, the word immunostimulation and inflammation is the same. So everybody here knows that immunostimulation is crucial to recognize and eliminate danger caused by microbes and tissue trauma. Importantly, inflammation is self-limiting and is attenuated after the elimination of the stimuli. Immunostimulation is crucial for restoration and homeostasis, crucial for tissue repair. So we start to have difficulties when inflammation is persistent, okay? Because persistent immunostimulation leads to the development of cancer, autoimmune disease, and hypersensitivity. The nature is well known. You have inhibitory process. We call it immunosuppression or anti-inflammatory responses. They are necessary to control inflammation because they contribute to down-regulate inflammation, and immunosuppression comprise key molecular mechanisms that control inflammation and inflammatory pathway. It's very important that since 10 years, we understand that a persistent immunosuppression not associated to inflammation can also lead to diseases. And we know that persistent immunosuppression favor infection, mainly in the lung, but also cancer. And I would like to that you keep in mind the two words here, immunostimulation and immunosuppression in mind. So what about toxicology and immunosuppression? You have often immunosuppression in the literature, in toxicology. I noted here two books or a review paper, and you can see that solvents, metals, biocides, all are related to immunosuppression, and we know also the molecular mechanisms leading to immunosuppression. But you can see here that finally the literature is pretty old, and often the immunosuppression is not well popular and not well described in the literature. And if you take the International Agency Research on Cancer, IARC, you have in the plan, that's a very good paper of Smith concerning all the characteristics of the carcinogen molecules or substances, and how to characterize the carcinogens. And you can see that in the list of the characterization or the characteristics, you have the point 6 and 7, where carcinogen induce chronic inflammation. And we know well that how to characterize inflammation is elevated leukocyte in the blood, elevated myeloperoxidase activity is related to the neutrophils. You have elevated chemokines and cytokine production. It's easy to determine. However, the point 7 is concerning immunosuppression, and it's a question: is immunosuppressive molecules? And you can see that you have the characteristic is related to a decrease of immunosurveillance, immune system dysfunction, etc. So it's not a positive but negative activity. And again, I would like to present a paper of Smith again, because concerning the PFAS activity. And you can see, and you follow the IARC categories, and you can see that again, it's easy to demonstrate the inflammation part, because you can note in the publication an increased cytokine production, etc., etc. But again, to characterize immunosuppression, we use decreased B cell activation, etc., etc. And you can see that immunosuppression definition is often related to a decreased function instead of an increased function. And you can see that all the characteristics here are related to inhibition of production of cytokines, inhibition of antigen presentation, inhibition of T cell function, phagocytosic function, etc., etc. So to characterize immunosuppression, it's often related to decreased functions. I like very much this slide, started a long time ago, to finally define and to present an historical progression of immunosuppression in the literature, because in fact, we have all now the elements to determine an immunosuppressive response. I would like to emphasize the fact that the first cytokines with immunosuppressive function is TGF-beta. So because we know that without TGF-beta, mice die from severe inflammation and autoreactive T lymphocyte. The same with IL-10. IL-10 is the second immunosuppressive cytokine discovered in the past during the 90s, and because the deficient mice develop enterocolitis. We know that we have different subtype of immunosuppressive cells, like macrophage. We know that they are not only inflammatory macrophage, we have immunosuppressive macrophage. The discovery of Sakaguchi in 2000 is very important, because among the T cells, he discovered a regulatory T lymphocyte subpopulation, and T cells control autoimmunity, but amplify tumor, reduce tumor immunity. And so these T regulatory cells can downregulate the immune system and then be immunosuppressive. It's not the end, okay? Every year we discover new molecules and new cells like IDO. which modified the tryptophan metabolization, which is very important. The tryptophan is very important for T cell activity. And we know that B cells can be regulatory, can be immunosuppressive because they produce a high level of IL-10. We have myeloid cells, immature cells. I will talk in a few minutes about this type of subpopulation. We have, of course, immune checkpoints. You know them because it's very important in immunotherapy, but I would like to finish this historical point of view by mentioning immunosuppressive fibroblasts, just to say that every year we discover a new population, and can be not only the immune cells, but also fibroblast epithelial cells, because they also are able to produce immunosuppressive mediators. And of course, the metabolism is also associated to immunosuppression instead of inflammation. Just to mention that if you learn or if you read about immunosuppression, is very, very popular since this Nobel Prize in 2018. Alison and Anjo, of course, discover the fact that you have immune checkpoints like CTL4 and PD-1. There are clear application in clinics, in the hospitals, to cure or to block cancer progression. But for me, it's important to emphasize the fact that they know that tumor cells manipulate a leukocyte and polarize them in immunosuppressive cells, which favor tumoral evasion from anti-tumor T lymphocytes. It means that immunosuppression produced by the cancer cells of the tumor can downregulate the T lymphocyte. And this point well illustrates the notion of immunosuppression. So I will now speak about our data, and just for this presentation, I decided to show you our history, our progression concerning the duo between inflammation and immunosuppression. And I will explain the reason why we are working on inflammation and immunosuppression. So the beginning of my story is around 2000, when we started to look at the effect of particles like silica, asbestos, and carbon nanotubes on the rats. So what we do, we inject in the lung the particles, and it's easy in this kind of animals. You induce rapidly an inflammation. This inflammation is persistent, characterized by macrophage, inflammatory monocyte, T effector cells, and inflammatory neutrophils. They produce growth factors and cytokines that explain fibrosis, and neutrophils produce oxidants that explain cell transformation. It is well known also that TNF cytokines can activate the cell proliferation. So the story is simple. We have a dogma concerning, the implication of inflammation during particle-induced fibrosis and cancer. So all is fine, all is simple, I don't think, because when you look at the clinical data, you can see that there is no evidence that steroids, the anti-inflammatory strategy, help the patient developing, for instance, silicosis or people exposed to particles. So clinicians are not sure that inflammation is the only way to induce fibrosis and cancer. And the second point is me here, because we observed several years ago that the mice, instead of the rat, develop a different responses, what we call disconnected There is a disconnection between inflammation and fibrosis. This is illustrated here in this graph. Particle-induced in mice inflammation, but the inflammation is not persistent. It's just acute. And then you have fibrosis in this animal. We demonstrate that this inflammation is not directly connected to or related to fibrosis, because if you treat mice here with steroids, strong steroid like dexamethasone, you block inflammation, but you do not block fibrosis. So it's very efficient to block acute inflammation, but not fibrosis here. In white column, you have the control treated mice treated with silica. You can see that fibrosis is not modulated by dexamethasone. The same if you take the MID80 knockout mice, if you block NF-kappa B signaling, you do not modify fibrosis. In contrast, if you use knockout mice for IL-10, you reduce the fibrosis. And at the beginning of this observation, you say, okay, between inflammation and fibrosis, maybe we have kind of immunosuppression, anti-inflammatory responses that block inflammation but produce fibrosis. And we observe the same in the silicotic patient. We go further and decided to say, okay, maybe during this phase, immunosuppressive phase, we have a different subpopulation of immune cells that regulate or orchestrate fibrosis. And what we observe is the presence of Tregs. It was a very impressive accumulation of Tregs instead of T effector cells. And we have demonstrated that this kind of Tregs produce PDGF, and the growth factor activate fibrosis. And you can see here on this graph that if you select Tregs from treated mice with silica, you purify them, and you inject the purified Tregs in the lung of naive mice, you increase fibrosis, you induce fibrosis as silica particles. And if you block PDGF, you reduce this activity. This is not the case with the T effector, the inflammatory T effector cells. So I would like to spend more time on new data that we observe in the lab. And the new data that we have at the moment is concerning the myeloid-derived superior cells. It's not a very new immunosuppressive population because we discovered this kind of immunosuppressive population, let's say 20 or 10 years ago. So the definition, keep in mind MDSC, and the definition of MDSC is myeloid-derived suppressor cells. MDSC are heterogeneous population of immature myeloid cells. What we call them monocytes MDSC and neutrophil MDSC. What Bronte and Gabrilowicz, they are the star of the MDSC, discover is the fact that those cells suppress innate and adaptive immune response by expressing suppressive factors. And now, today, MDSC are important in cells, in cancer, and in infection. So there are important cells in cancer and infection. So you have some application, and we know more and more concerning MDSC. In the beginning of this project, I was in fact contacted with a colleague, a Belgian colleague, Van Ginderachter, Joe, in a university in Brussels, close to me, my university, and he discovered the fact that you can, in mice, detect MDSC by using some markers like CD11B and GR1 positive cells. You can differentiate this population in two subpopulations, LY6C for the monocyte MDSC and LY6G for the neutrophils population. But he discovered that the presence of this kind of MDSC is their ability, okay, to neutralize or to suppress T cell function. And that's is well illustrated in the review of Gabrielowicz, where he showed that the neutrophil and the monocyte MDSC has a main function is to stop the T cell proliferation and activation by releasing different markers or molecules coming from arginase, PG2, TGF-beta, IL-10, IL-10. And so, but you can identify them, but to demonstrate that if you have this population, you need to use immunosuppressive tests. This immunosuppressive test is performed by combining the immune cells to test monocyte or neutrophils to activated CD4 T cells. We use in vitro antibodies directed against CD3 or CD28. Those antibodies will activate the proliferation of CD4 and CD8 T cells. So you do a co-culture and you look at the proliferation of lymphocytes. And you can see that this is the first paper mentioning this bioassay to detect MDSC. You can see that when you combine monocyte or neutrophils that have an immunosuppressive activity, you reduce, you inhibit the proliferation of T lymphocytes. And what we did during our research is to purify monocytes and neutrophils. This is first neutrophils. We purify them from the lung of silica-exposed mice. And you can see that, of course, the cells, the monocytes, are different between the different conditions. They are not exactly the same. And we also purified the monocyte coming from the blood to have a negative controls. And we decided to associate those purified cells with T cells activated with the antibodies. And what you can see that if you take monocyte from the blood, from a mice, you can see that you don't have any effect, meaning that the monocyte from the blood do not deactivate or do not inhibit the T cell proliferation. It's the same when you take the monocyte from the lung of saline, a control mice. You can see that even at a ratio of 1 T lymphocyte and 2 purified monocytes, you don't have any activation, any effects in the co-culture. In the contrast, if you take purified monocyte from the lung treated with silica, you can see that the monocyte has the capacity to downregulate the proliferation of T cells in vitro, meaning that indeed, in the lung, you have monocytes, and this monocyte can be immunosuppressive and can be defined as MDSC, monocytic MDSC. We demonstrate that they are immunosuppressive because they produce arginase. It's just a remark, it's not very important, but we demonstrate that the activity is completely inhibited if you use blocking or if you block arginase here, demonstrating that, okay, we have a subpopulation only in the lung treated with silica. And this immunosuppressive activity means related to arginase, means that you have MMDSC in the lung. What are the function of the MDSC population? We demonstrate that MMDSC activate lung fibroblasts, again in co-culture system, where you can see that monocytes obtained from the lung of silica-treated mice, but also nanoparticles here, fibrogenic nanoparticles, you can see that they induce the production of TIM1, which is a marker of fibroblast activity. And this is an inhibitor of metalloproteinase, which inhibit the degradation of collagen. And this activity is significant. And we know also that MMDC produce or express TGF-beta. And if you block TGF-beta in the co-culture, you block the activity of monocytes in the production of TIM1, demonstrating that again, MMDC are immunosuppressive, but they have other function. They have also the possibility, the activity to activate fibroblasts, at least in co-culture. So what we did is, of course, to block the recruitment of MMDAC in the lung by using the CCR2 knockout mice, and you can see that you reduce the development of fibrosis after silica treatment. We were very happy to see that in lung fibrosis in human, you have this kind, you have also this kind of immunosuppressive MDSC, demonstrating that it's not only in the mice system that you have this subpopulation. So we go further and decide to investigate neutrophils, because the question was, okay, maybe in some system you have the recruitment of inflammatory cells, the famous neutrophils, but they are not inflammatory related, but they are more immunosuppressive related cells. And for that, we took the same strategy than before. I mean that we purified neutrophils from the blood, from the lung of control mice, the lung of silica-treated mice, but also we took the neutrophils from mice treated by LPS. And LPS induces a classical neutrophilic inflammation. And we perform again the in vitro test combining T cells and neutrophils. And you can see that, as expected, blood neutrophils do not induce, do not affect the proliferation of T lymphocytes. The same with the neutrophil provided by the lung of saline-treated mice. But if you take those neutrophils coming from silica-treated mice, you can see that again, in function of the number of added in the co-culture, you have a reduction of T cell proliferation, and meaning that again, in the silica-treated mice, you don't have inflammatory neutrophils, but instead you have neutrophil MDSC. Importantly, if you take the neutrophil coming from the LPS-treated mice, you don't have this activity, again, showing that this population is specific to silica-treated mice. We just demonstrated that this population of immunosuppressive MDSC is present in silica, of course, but is present also after installation of CNTs, the carbon nanotubes. Those particles also induce lung fibrosis in mice and in rats. We show that we separate the neutrophils to T lymphocyte, and we demonstrate that just the neutrophils release an inhibitor is not a cell contact that explains the inhibitory effects. And we demonstrated by using antioxidant molecules that the inhibitory effect of neutrophil MDSC is related to the release of ROS. So the classical activity of immunosuppressive neutrophilic MDSC. So, of course, in the context of lung fibrosis induced by particles, we decided to look at the gene expressed by those cells. And we, of course, demonstrated first that the MDSC obtained after silica treatment are completely different to the inflammatory neutrophils observed after LPS injection. You can see here the expression of neutrophilic granule protein, NGP. You can see that neutrophils purified from silica-treated mice do not express NGP, in contrast to the neutrophils obtained from LPS-treated mice. The same with the CXCL1. We know that this chemokine is well expressed by the inflammatory neutrophil, which is not the case when you consider neutrophils purified from silica-treated lungs. We searched a lot concerning different genes, and we were impressed by the fact that neutrophilic MDSC express specific genes related to macrophages, because we demonstrate that osteopontin was overexpressed in the neutrophilic MDSC, in contrast to the controls, but in contrast also to the LPS-treated cells and the lungs. It was the same for the MCSF. We noted a specific overexpression, meaning for us that, okay, those neutrophils, immunosuppressive neutrophils, are maybe related to macrophage population. And it's the case. We use an antibodies neutralizing the neutrophilic MDSC in silica-treated mice. And what we observe is a complete decrease in the number of course, of neutrophilic MDSC, but also the number of alveolar macrophage. You can see here that you have few fresh macrophage. It's more foamy macrophage with a lot of silica particles. So it means that the neutrophils, MDSC, in silica context, help the recruitment of macrophages to the lung. So for this conclusion, and this is a better view of what we observe since 10 years now, we noted in a model that finally we have immunosuppression. We determine the immunosuppression by identifying the different subpopulation of M2 immunosuppressive macrophage, Tregs, immunosuppressive T cells, and immunosuppressive neutrophils and monocytes. They express different chemokines, cytokines, and growth factors that explain fibrosis. So it was the first demonstration that inflammation is not all, and that immunosuppression can also participate in the development of fibrosis in the cancer. Concerning the cancer, we have, and I have 2 slides to explain the same strategy in another model. It's a more cancer-related model, to demonstrate that this kind of population, immunosuppressive population, is not only related to fibrosis, but can be also implicated in particle-induced cancer. So what we did here is use the rats, and we injected fibers like asbestos and CNT7, and that induce mesothelioma, and particles, CNTT, that do not induce mesothelioma, to compare the capacity of the cells to be implicated in the mesothelioma process. You can see here that in this model, you have a clear mesothelioma development. Why we use peritoneal cavity? Because it's more easy, it's more quick than the injection into the lungs. So we observe again different subpopulation, and we were impressed by the fact that macrophage present in the cavity of the treated rat can be or are immunosuppressive, because if you combine those purified monocytes coming from treated rats, they are able to inhibit the T cell proliferation in vitro. And so we say, okay, we have again a new population of immunosuppressive cells. And we were very happy and decided to look at the different immunosuppressive, because we have an activity, but we need to determine the production of immunosuppressive mediators to demonstrate the activity on the T cells and on other cells. And so like before, we try to identify very well-known immunosuppressive molecules like TGF-beta, IL-10, PGE2, arginase 1, and more and more. And we were surprised that in any condition, those immunosuppressive macrophages produce TGF-beta, IL-10, or PGE2, or arginase 1. And we were very surprised because no very well-known immunosuppressive molecules were present or expressed by our macrophages. So, like everybody know, we did RNA-seq approach, RNA-seq approach, and we observe and we selected the gene which is only expressed by the macrophage coming from the mesothelioma model. And it's very important to have these comparative models to select genes or molecules related to the cancer process. And you can see that effectively, macrophage coming from the CNT7 treated rat overproduce syndicon-1. So syndicon-1 was the mediators overexpressed in this condition, but not in the control condition or in the condition where you have a treatment with particle, but you don't have any mesothelioma development. It's the same here concerning the protein, it's the same with asbestos. And we measure the protein, but also the gene. We have localized the expression of syndicon-1 in the tumors, and we demonstrate that only macrophage express syndicon-1. So we don't have, of course, the end of the story because we are working on, but this data illustrate that the fact that we are not in the end of the immunosuppression, we will probably discover several additional immunosuppressive cells, but also immunosuppressive mediators. So I am in the end of this presentation, and I would like to combine everything, and I would like to combine the magic words, inflammation and immunosuppression. I think that it's clear that inflammation can induce cell injury and TC damage, initiate cell transformation, orchestrate leukocyte accumulation, but also produce a cell growth environment to activate fibrosis and cancer. I am sure of that. But my idea is to support that beside inflammation, if you have immunosuppression, you can also induce diseases. Why? Because immunosuppression can control exaggerated inflammation, but prevent T cell response. And you know that in the cancer, it's a key issue, but produce also a cell growth environment like inflammation. And you combine both, inflammation and immunosuppression can so induce autoimmunity and cancer and fibrosis after the treatment of different particles like asbestos, silica, and carbon nanotubes. So this is my last slide. So just few perspective, few ideas about this new scenario between inflammation and immunosuppression, immunostimulation and immunosuppression. I think that this new scenario explains the diversity of the disease induced by particles, but probably also induced by other substances. This scenario explains why existing therapies are disappointing with differential effects in patients. The scenario helps us to select the most appropriate therapy, I am talking about the clinician. They don't know if they need to treat with immunosuppressive steroids, immunosuppressive compounds, or apply the same strategy than in immunotherapy. And I think that is very important in immunotoxicology, in toxicology in general. We need bioassay to determine the activity of pollutants, but also the new drugs or the new substances. And I think that the notion of immunosuppression, considering the cell identified yet, now could be enrich our view or our current bioassay testing new substances. Okay, I think I am on time. I just would like to thank a lot of people, of course, in every lab in the world, the technician, but also the postdoc and the PhD student. They lead all the studies and they produce all the data that I have shown today.
Thank you, François. This was excellent and very nice and original research. So I have already some question. I have a question from Gad Olivier. So my question will be, I have two questions from two questions. So how do you see lung microbiota dysbiosis part of the immunosuppressive impact?
Yeah, excellent question. So to be honest, we try to identify microbiota in the lung, and we didn't see any difference. However, I think about the metabolites produced by the microbiota, and I am sure it's well accepted in the immunosuppression world that the metabolite can be transformed, differentiate the cells from inflammatory state to the anti-inflammatory immunosuppressive state. So I will not be surprised that in the future, metabolites transformed by microbiota or the cells should or will modify the immune profile of the cells into the lung. But at the moment, I don't see clear data showing that the microbiota can directly transform the immune cells or polarize the immune responses.
To go to the second question, as mitochondria can be impacted by immune system dysregulation, do you think that mitophagy and mitochondrial renewal could positively impact the lung immune system in allergy and disease?
Yes, I think that's the key issue. What we know from our side is the fact that so when I talk about persistence responses, okay, you need a response, a long response with the same pathway activated by, and I think that this kind of pathway can be also implicated in the differentiation and the persistency of the responses. However, when we look at the macrophage at the moment, we try like everybody to block this kind of response. So we didn't see any sign of activity concerning the macrophage differentiation, only the macrophage differentiation and the macrophage activities, immunosuppressive activities. And I think what we observe, the negative data that we have, is because maybe we look only at the classical immunosuppressive component, like IL-10, TGF-beta, and PGE2. But I am sure that if we repeat some experiments with different markers, because we realize that with all the work that we have done, it's the fact that, okay, you can not be limited to the classical immunosuppressive mediators. You need to look at, for instance, interleukin-1, and so the panoply of immunosuppressive mediators can be different, but at the moment I don't see a clear effects.
So now we have a question from Thierry Claire. So does the MDSC phenotype kind of markers such as CD11B or cytokines are specific to the kind of fibrosis perpetrator, meaning, for instance, immune cell signature specific to CD4 only?
I propose to read the paper of Gavrilovich, published in Nature, and he proposed new biomarker, CD markers for the MDSC. And of course, you can have different markers to define the MDSC population, but at the end, you need to prove that they are immunosuppressive in vitro. And that's the difficulties to work with MDSC. Concerning the question, I think it seems specific to particles, but we only tested LPS instead of particles. So, and we don't have a clear control situation. I mean, if we use other particles like inert particles, they don't induce inflammation, so you don't have neutrophils from those kind of situations. So we took only positive controls like LPS, and what we observe is the fact that if you have in the mice system particles inducing fibrosis, you have this kind of subpopulation. What I would like to say that in the context of the particles, often the biomarkers of toxic particles is the number of neutrophils. And if you take these concepts, you can have inflammatory or immunosuppressive neutrophils. So the count of neutrophils is the same. So it's still a good markers of toxic particles in the lung. But it is interesting to say, okay, maybe behind these neutrophils, you can have different activities. What we did not, it's the nanosilica particles. What we didn't test the neutrophils coming from nanosilica. So it's another possibility, but we didn't do the job for the nanosilica.
So I've got two questions related to this. There's a question from Amandine Lay, and she wants to have an idea of the size of the particles you used. Mm-hmm. And a question from Armel de l'Avidamment that is, she wants to know if you have used amorphous silica in your system.
So we didn't use amorphous silica. It's a good idea. And the size silica is not nanometric, it's just micrometric. So okay, I don't have the data to conclude about nanosilica. Sorry for that.
And now we've got a question from Julia Filipovska. So it is fair to conclude that there is a bad, which is syndecan, I guess, and good, interleukin-10 and alike, immunosuppression. But this is a question mark. If so, it would be important to understand what drives one of the other immunosuppression once inflammation is induced. That's a good question.
Good question. To be honest, what we did at the moment is the fact that we try to block immunosuppression. Okay. I will not enter in the details, but the Treg story is very interesting. If you block Tregs, you block Tregs, you increase inflammation. So it's a balance. So at the moment, if you modified one part of the balance, you increase the other one. So it's always, we are in the end because we have no additional IDs, because if you modify the inflammation, you have immunosuppression. If you modify immunosuppression, you modify inflammation. So at the moment, our best strategy is to, that's the idea, is to block one immunosuppressive mediators like Sanecan-1. But we don't know whether we can also modify the inflammation, because if you remove immunosuppression, we will have much more inflammation. So it's a complex situation. Sorry.
Thank you. I have a question from Sadia Karen-Romer. In fibrosis, do the Treg crosstalk with the MDSCs? And if yes, is it the mechanism of the regulation of inflammation through Th17, or for instance?
Mm-hmm. Good question. Difficult question. We don't know. Just what we know is the fact that the Tregs produce a high level of PDGF, okay, and TGF-beta. So I suppose that maybe Tregs can effectively support MDSC, but I am not sure. I don't know the connection between the both cells. It's a good idea because I suppose when you see the whole population present in during fibrosis, you have macrophage, of course, T lymphocytes, monocytes, and neutrophils. So, and at the same moment, at the same situation. So I suppose that probably you have a cross connection between the different subtype of cells.
So I have the same kind of question to follow on, is that, did you look at the kinetics of lung infiltration by the different type of cells? I mean, the Tregs are coming first and then the MDSC, or?
No, they are coming, in fact, it's very simple. So at 3 days, you have mainly neutrophils, inflammation, etc., monocyte. And then at 15 days, after silica treatment in the mice, you have the recruitment of Tregs, but neutrophils, monocyte, and macrophage. So you have always the same subpopulation after 2 weeks, meaning that you have a first step, which is inflammation, the classical inflammation. Probably the inflammation induces an anti-inflammatory immunosuppressive response. And this response is characterized by a mixture, a mix of immunosuppressive cells, comprising T cells, monocytes, exactly the same population that you have during the inflammation process.
But when you say they are Tregs, okay, when you say they are Tregs, you say that these cells are producing PDGF and TGF-beta, okay? So they are conventional Tregs, or there are other subtypes of Tregs? Because usually, when they are producing also interleukin-10, these Tregs, or yes?
Yes, we didn't identify Tregs, our Tregs in silica-treated mice as a high producer of IL-10. Okay, so we don't have any data concerning the characterization of the Tregs. Well, what is missing during our experiment is to compare our Tregs with other type of Tregs, even in vitro. And at that time, concerning the experiment, it was not possible to select the two subtype of Tregs. So it's difficult for me to answer the question, but I suppose because they don't produce IL-10, etc., they are more conventional. They are probably conventional instead of different.
Okay, so okay, it's time. Last question, it's okay. I have a last question, short question. Do you have any hypothesis how the Tregs are attracted to the lung? Okay,
so that's we think that it's a chemokine cocktail. Okay, but the other, and I don't have the answer, chemokines can be important for the Tregs recruitment. So, but what we know also is the fact that they can also proliferate, okay, because so it's not impossible that in the context of immunosuppression in the lungs of silica-treated mice, we have a kind of proliferation. It's surprising data because Tregs are anergic cells. But and it's the reason why probably, so we identify the Tregs by measuring FOXP3, but we didn't do the immunosuppressive test with the Tregs. And so I am not sure that it's the complete Tregs population, but we can have T cells, immunosuppressive T cells as well.
Okay, so it's time now to finish. The time is over. So thank you very, very much, François, for this excellent presentation. It was very informative, very interesting, because you have a broad picture, and then you did go in the detail of the mechanism. So thank you again. The next webinar will be in April, probably the week of the 21st of April. And it will be given by someone from the Early Career Forum from EUROTOX. So it will be an early career scientist. And will be posted. You will be posted. Thank you for attending. We had 50 people to date, which is it's a nice attendance. So thank you very much, and thank you for the question. Thank you.
Okay.
Bye-bye.
Bye. Thank you. Bye.
Bye.